Hello students, our chapter is chapter number 1, an introduction to drugs, their action and immunobiological. We already have discussed about vaccines, introduction of vaccine, method of vaccine production, like there are 3 different methods of vaccine production. 2 methods we have discussed, that is first one is inactivated and killed pathogen method and second one is live attenuated pathogen method. This one is a second method of vaccine production. Now today we will discuss the third method of vaccine production. So what is that? That third method is cellular antigen from a pathogen. This is third method of vaccine production. The essential component, there are some essential component for vaccine production. There are some components like is one or more antigens. Sometimes more than one antigen is required for vaccine production that induce immune response and that provides protection against any disease. Okay. Essential component of this type of vaccine is one or more than one antigen that induced immune system or immune response that provide protection against any disease. The surface antigen is harvested from the pathogen. There is a surface antigen which is harvested from the pathogen, then it is purified and reconstituted into vaccine preparation. So these antigens can take a number of forms, including the carbohydrate capsules as in Neisseria meningitidis. There is Neisseria meningitidis pili. What is that Neisseria meningitidis pili? It is gram-negative bacterium that can cause meningitis and other form of meningococcal diseases. There are different meningococcal diseases such as meningococcemia. Meningococcemia is a life-threatening sepsis. It is like a septicemia. It is life-threatening sepsis. Okay. So, Neisseria meningitidis pili and the viral protein coat as in the vaccine for hepatitis B. So, they also called as acellular vaccine. These type of vaccine are called as acellular. What is acellular vaccine? We are discussing about cellular antigen from a pathogen. But in the case of Neisseria meningitidis pili and the viral protein coat, as in vaccine for hepatitis B, they are also called as acellular vaccine. So, acellular vaccine means a vaccine that may contain cellular material but does not contain complete cells. So, that type of vaccines are acellular vaccines. Some other examples of acellular vaccines are pneumococcal conjugate vaccines. There are pneumococcal conjugate vaccines are also example of acellular vaccine which does not contain complete cells. For example, PCV7, PCV10 and PCV13 are examples of pneumococcal conjugate vaccines and vaccine against Haemophilus influenza type B, that is HIB, Haemophilus influenza type B vaccine, and next one is a human papilloma virus, that is HPV, human papilloma virus, which causes warts all over the body, and next one is a whooping cough. So there are some vaccines which are also called as anticellular vaccines. Okay. So there are some advantages of this method. This method is cellular antigen method. This method is cellular antigen method. So there are some disadvantages of that method. There is virtually no chances of infections by these vaccines. This method, like cellular antigen form of pathogen method, don't have virtually no chance of infection by these vaccines because the antigens do not have the capability of reproducing themselves. Antigen don't have capacity to reproducing that dangerous diseases or infections. Okay. Thus, the immune response in case of vaccination is not very aggressive and their storage is easy and there is practically no possibility of contamination or reversion, because the storage is easy and storage is easy of that vaccine, and there is no possibility of contamination or reversion. Okay, so there are some challenges of this cellular antigen from pathogen. There are challenges or disadvantages. Correct identification, characterization and purification of antigenic components for the sake of consistency and safety is the biggest challenge. In the case of this type of vaccine, cellular antigen from pathogen type of vaccine, correct identification, characterization, its purification of the antigenic component for the sake of consistency and safety is great challenge. So pathogen has to be grown under carefully controlled conditions. Okay. Then these antigen vaccines are strain specific, and characterization of new antigens from different infections or different infection models makes it necessary to upgrade the existing vaccines with new antigen. For example, in gonorrhea may require 1500 different peeler antigens. Okay, so this one is an example. Cellular vaccines may exhibit lower antigenicity in the way. So when that type of vaccine shows lower antigenicity in very young, and that's why that vaccine may require several injections for full immunobiological competence. Okay, nowadays antigens are available to trigger immunoresponse and provide protection against hepatitis B, hepatitis C and our today's coronavirus, that is COVID-19. Okay, so there are some advantages of vaccines. In general, what are different advantages of vaccines? That there is virtually no chances of infection by this type of vaccines, because the antigen do not have the capability of reproducing themselves. This one is great advantage of cellular antigen from a pathogen. And the immunoresponse in case of vaccine is not very aggressive. This is another one advantage of this type of vaccine. Okay, and the storage is easy and there is practically no possibility of contamination or reversion. This is third advantage of this type of vaccines. So today we have discussed all this third type of vaccine preparation method, that is cellular antigen from a pathogen. So now we will discuss general advantages of vaccines. Okay, so there are some advantages like vaccine. Why we use vaccines, or what are advantages of vaccine? It gives protection of population against infectious diseases. It shows protection of population against infectious disease. Then second one is prevention of spreading of epidemic and pandemic disease. Then third one advantage is prevents disease spreading to others. This is third one advantage of this vaccine. Vaccine avoids large cost for the treatment of infected patients by avoiding infection. When we use vaccine, it avoids large cost for the treatment of infected patients by avoiding infections. Okay, there are shortcomings of vaccines. May have some side effect. That vaccine may have some side effects. Sometimes it is severe, sometimes it is normal, or not severe type of side effects. Vaccine requires National Health Service individual outlets. It requires National Health Service or individual outlay. Then uncertainty about complete protection. Vaccine shows uncertainty about complete protection. Okay. Then sometimes injections are not pleasant. Normally injection or injecting vaccine or vaccine injections are not pleasant. more immunobooster injections are inconvenient, and more injections, more than 1 or 2 injections or boosters are normally inconvenient. So these are shortcomings of vaccines. Okay, now our next point is SARS, COVID-19. COVID-19 is pandemic. In last 2 decades, coronaviruses caused 2 pandemics, namely severe acute respiratory syndromes, that is SARS. What is SARS? Severe acute respiratory syndrome during 2002 to 2003, and Middle East respiratory syndrome, that is MERS, in 2011. Okay, these two pandemic caused by coronavirus. The December 2019 outbreak of coronavirus originated from Wuhan, China, is called as novel coronavirus disease, that is novel coronavirus disease 2019 or COVID-19. In the early period of this pandemic, almost all the countries were caught unaware, and the number of cases increased drastically. As per guidelines, restrictions on public gathering, public transport system, schools and colleges, shopping malls and entertainment centers were implemented. Lockdowns was enacted in the region with higher number of cases to break the chain. Then wearing of masks in public places and use of sanitizers was strongly recommended as the first line of defense against the spread of COVID-19 infection. According to the International Committee of Taxonomy of Viruses, that is ICTV, the coronavirus are subclassified on orthocoronavirinae, which in turn consist of 4 categories: alpha, beta, gamma and delta. There are 4 categories of coronavirinae, which in turn consist of 4 categories, that is alpha, beta, gamma and delta. The alpha and beta coronavirus transmits diseases in mammals such as bats, pigs, cats and mice. Bats, alpha viruses transmit disease in mammals like bats, pigs, cats and mice, while gamma and delta COVID virus usually infects birds. The beta coronavirus class includes SARS COVID. SARS means, that is severe acute respiratory syndrome, and MERS COVID like Middle East respiratory syndrome, and COVID-19, or this is SARC means severe acute respiratory syndrome, that is COVID-2. Its genome is fully sequenced. The genetic sequential arrangement of SARS CoV-2 has an identical genomic array of SARS. It attacks the lower respiratory system to cause viral pneumonia. That COVID-2 attacks the lower respiratory system to cause viral pneumonia, but it may also affect the gastrointestinal system, heart, kidney, liver and central nervous system, leading to multiple organ failure. As per World Health Organization data, worldwide 22 crores 88 lakhs 7,631 confirmed cases of COVID-19, which including 46 lakhs 97,099 deaths have been reported till the 3rd week of September, out of which the second highest number, that is 3 crores 35 lakhs 4,534 of reported cases are from India and USA. So these are some details about SARS, that is severe acute respiratory syndrome, SARC COVID-19 viruses. Okay. Now in our we have already discussed about vaccines, there are different methods of vaccines. Its introduction, then vaccine production method. There are 3 different methods. And our today's lecture, we have discussed about cellular antigen from a pathogen method, advantages of vaccines, and some shortcomings of vaccines, and what is SARS COVID-19. Okay, so in our next lecture, we will discuss treatment of COVID-19 infection and vaccine against COVID-19. Okay, thank you.